Okay, so sir has already given a generalized definition, which may be common to all. But if you find in Oxford Dictionary or some big book like David Mount Bioinformatics or Pevsner's Bioinformatics, so you'll be getting definitions like this. So it will be the same thing as Soromonta sir has generalized already. So somehow in Oxford Dictionary, it is elaborately mentioned as bioinformatics is conceptualizing biology in terms of molecules in the sense of physical chemistry and applying bioinformatics techniques to understand and organize the information associated with these molecules on a large scale. So in short, bioinformatics is a management information system for molecular biology and has many practical applications. So history begins with this fellow, that Mendel, when he had performed his experience. Data generated at that time itself, and management of data was required at that time also. Then much more data generated when DNA discovered in 1969, 1869. 1944 demonstrate that DNA is the major carrier of genetic information. Then Watson and Crick, most of you know that they have discovered the 3D structure of DNA. So informations are generated due course of time, and we need much more sophisticated management system. So like that, you can see in different phases of history, different things have emerged out. So in 86, latest discovery of RNA splicing. So and still RNA splicing is the alternate method for finding important genes and all. Human Genome Project, that in all of us the same thing is going on, but we are different. So this is all because of the enormous number of data generated in due course of time. So in future, you can see that 10 to 20 years that Genomes to Life project will come, ENCODE project. These new projects will be coming up. Okay. So this is this bioinformatics. Okay, it's fine to say as an interdisciplinary subject. So far as most of the discipline like physics, chemistry, even mathematics, statistics also can be used to analyze biological data. But something is called transdiscipline, which is not multidisciplinary. Okay, so transdisciplinary is that discipline, that subject in which you have to apply and you have to read or you have to conceptualize the method described in the textbook. If you see in this figure, the biology, computational biology will fall in this region. That means people who can acquire knowledge of analyzing sequence data. So then structural bioinformatics, database management, database uses, then systems biology framework. They are called as computational biologists. Okay. So computational biology is a transdisciplinary subject, which implies that you have to apply the all relevant field, statistics, mathematics, physics, chemistry together at a time. Okay. So this is a well-known figure, or most of the molecular biologists or the people who are from life science background, the central dogma scenario, which uh, itself is a data generating process. So you know, all of you, I think you know about this one. So when DNA transcribed to mRNA and mRNA gives rise to the protein, the entire process will give us a lot of data. So importance of bioinformatics, why it is important? To improve the ability to interlink the biological data. This can be used for revalidation studies. So interlinking is the main importance, that we are generating data. This generated data should be used by the other fellows also in different part of the world. Then improved gathering, management, storage and retrieval of the biological data. So we need to have a management system or a storage system or a process of downloading this data. So we need to get some new addition. So bioinformatics applications will be helpful for making this kind of system. And With the ease of bioinformatics, we can easily analyze the high throughput analytical by using high throughput analytical techniques. We can analyze the biological data in very fast manner. And designing tools for this is another importance, that there are many tools, many softwares available over the internet for analyzing biological data or doing different kinds of things in biology. But still, some are missing. So that can be identified only and only if you explore yourself what is missing. So that is the designing. That is the part of designing in bioinformatics, designing tools for identifying new drug target, creating new genetically modified food, diagnostic kit, and many more tools can be designed by uh, the knowledge of bioinformatics. So areas are most commonly the entire genomics and proteomics studies are included in bioinformatics. But in genomics, what we do is the study of the sequences, genomic sequences, gene organizations, mutational studies, studies at DNA level, then the study of information flow within a cell like functional genomics and structural genomics. In proteomics, uses the information determined by biochemical crystal structure method, like visualization of protein structures, then protein-protein comparisons. Then we can use the bioinformatics applications to determine the conformational changes, protein folding problem, and antibody design, antibody binding sites. We can find out, we can predict protein-protein interactions. We can predict, and we can give some helping hand to the real-time drug discovery process, which is itself is called as your computer-aided drug design. There are mainly three aims. That is, first and simplest, the bioinformatics organizes data in a way that allows researchers to access existing information and to submit new entries as they are produced, and to develop tools and resources that aid in the analysis of data. Okay, so this is somewhat tough for the biologist, but who can explore their career in biology as well as in computer science, they can fit into this. They can be fit into this. Okay, so to use these tools to analyze the data and interpret the results in a biologically meaningful manner. So this is the main aim of this program, of this training course, so that some of the tools will be given to you as introductory tool to be used in data analysis, biological data analysis. So we can use existing tools to analyze already existing data. These are the three main major aims in bioinformatics. So what are biological data? So basically, biological data, we talk about sequences, nucleotide sequences, DNA, mRNA sequence, tRNA, maybe ribosomal RNA also be a data. Then protein sequences, that is amino acid sequences. Then protein usually have got structures, and structures are usually determined by definite method. So 3D protein structures we have in some databases. So these are again types of data. Then complete genomes and maps, gene expression data, genetic variations, polymorphism data are also there. And now the emerging thing is next generation sequencing data. So it's a huge data. So we'll be discussing in due course of the classes. So what are the characteristics of biological data? So I'm giving some examples, simple examples from mostly known, mostly explored database, composite database, and CBI. So from there, I am giving some example. You see, the characteristics you can we can termed as the biological data are heterogeneous. So heterogeneity is the main uh, uh, characteristics. You see, I have taken some pictures of all 22 plus XY chromosome from human genome. So you see the length of the chromosomes are not same. Simple understanding. So data inside this, you just for the time being, you know that this data for chromosome number 1 will have some data, chromosome number 2 will have some other data. That means chromosome number 1 and chromosome number 2 will not have the same data, right? So they are heterogeneous. And by the length of the chromosome, you can easily understand that their data are not uh, equally distributed, or not two chromosomes are having same data. So they are heterogeneous, right? So next characteristic we can see, each and every chromosome, if you click, no, and see in NCBI, if you go and type human genome in the genome database, and you will see this figure of chromosomes, and each and every chromosomes will be clickable. You can click and see where I have clicked here. I have clicked here Y chromosome. So you can see chromosome number Y is having some data which is corresponding to OMIM. OMIM is online Mendelian inheritance in man. So it is pertaining to some disease causing gene. Okay. So how many genes, how many disease causing OMIM data, what are they means they are very complex. Okay. So if you want to see each and every data, you just have to click. So by clicking and clicking, suppose we have some data here. How much data we have? 1, 2, 3, 4, like that. Suppose we have 15 data up to this much. So 15 data, if you click one by one, it will not be helpful to you. You have to have some proper objective why you are exploring Y chromosome. So the characteristic, next second characteristic, is said to be as complex because data are arranged in such a way that we need to define something prior to any analysis. So that is why biological data are complex. Next is variable. Simple, again, simple figure depicts that chromosome number 1 to 5, and each chromosome number 1 to 5 are having different what proteins, protein encoded up to date. Then size MB is given. MB, don't be confused with megabyte. It is megabases. Okay, nucleotide bases. Then GC content, you can see GC percentage. So don't think this will be a constant. Today you are observing that chromosome number 1 is encoding 9684 protein. So tomorrow it will be 86, 9686. So then day after tomorrow it will be 10,000. So people are depositing data, and thereby their relevant data, their corresponding data is varying day by day. So variable. These are not constant. Biological data are not constant. They are variable. Okay. So biological database are depending on their features, whether we are considering sequences, we structures, or literatures. Biological databases are categorized in three categories. So let us have a look on this. So this is NCBI, National Center for Biotechnology and Information. This is not primary, not secondary biological database. This is a composite database. Composite database means it is having link to the other primary, secondary databases, and it itself is having storage, a huge storage. So you can see the drop down box. No, this drop down box is for you to choose the database. So if you want to type something here, suppose hemoglobin. So hemoglobin Homo sapiens, or hemoglobin human, or human hemoglobin. And if you are looking for the protein, you need to change this database to protein. Okay, you will have the drop down list. So this we will see in practical. Okay. So biological databases, roughly you can have a figure like this. So we have structure, sequence, and other data. We may have maybe literatures. So structure databases are kept primarily in protein data bank. Those structures which are determined by X-ray crystallography, nuclear magnetic resonance spectroscopy, then cryo-electron microscopy, this determination method. So determined structures are kept in protein data bank. Then depending on their architecture, like how they are arranged, whether the protein is only alpha helical, means only the alpha helix are present, then it may be categorized into alpha protein. If it is beta sheet containing protein only beta sheet is there. So it may be kept in your beta protein. Like that, scope is there, structural classification of protein. Cat is another one, depending on their topology and architecture, whether they are homologous or not. So structure already determined structures are kept in other databases. So there are many databases in sub databases in structure database also. I have listed a few only to give a gist of my biological databases. So sequence databases are majorly defined in two categories: protein sequence database and nucleic acid sequence database or nucleotide sequence database. So nucleotide sequence databases are constant. These three are primary nucleotide sequence database: GenBank, DDBJ, EMBL. So nowadays this EMBL is known as ENA, European Nucleotide Archive. The GenBank is NCBI itself. So DDBJ is maintained with your Japan. Again, protein sequence databases, SwissProt or UniProt is the primary protein sequence databases, and this contains the refined sequences. Okay, any protein sequences obtained from SwissProt, UniProt is likely to have less error, means or no error. Okay, so PIR is another one. TREMBL, translated EMBL, is a computer annotated database. ProCite, PFUM, these are the protein sequence databases. So there are many databases. Okay, I have not listed all. So only for idea, I have created this figure. Okay, so above that we have some more like pathway database. Okay, managed information related to cellular pathway, metabolic pathway, enzymatic pathway. We have CAG, ligand, Brenda, many, not very many, but these are few, again, pathway database. We mostly, when we have to deal with some pathway, we mostly see in CAG. CAG is your Kyoto Encyclopedia of Genes and Genomes. Okay, so you can explore. You just type in Google CAG and see at the page, and in that page every website have a search box, so you can search any pathway. You search glycolysis, gluconeogenesis, apoptosis, insulin signaling pathway. You will see the entire pathway with hyperlink to each and every biological entity. That may be either a protein or gene, if it is functioning, or if it is acting. Then you can see those information about genes or protein with the hyperlink linked to those entries. Okay, so this is the look of the CAG pathway database. So you can search by the pathway name, enter keyword here, and get the pathway. Just explore it. You can see that. So we'll be exploring this on practical sessions. So there are many tools. I am just giving some figure to depict that there are many tools. The ocean of tools are there. So we cannot make a comprehensive list for bioinformatics tools. Okay, so there are many tools depending on the uses. So we'll be discussing that. So let us let us have a quick look on the biological sequence analysis. Why we should do biological sequence analysis and what is it? Okay. So sequences. By now, I think you are having some idea now. What are the sequences? So sequences. When we talk about nucleotide sequences, it is nothing but a long, repetitive ATGC letters, right? Then, if we say about amino acid sequence, then it is the one-letter code of all 20 amino acids repeatedly arranged in indefinite way. Okay, so sequences related by evolution, right? So when we say that this evolution term, no, that immediately next thing comes in our mind that if they are related by evolution, they may have common ancestor. They may not have, but common ancestor term is coming, or they may be modified over time, and this modification may be of biologically relevant changes, maybe in a single residue mutations in case of your polypeptide chain, that is in protein sequences, or maybe deletion insertion in the segment, right? And suppose, for example, we have this four sequences, right? So four sequences, if we assume that these four sequences are from same protein of different organism, uh, suppose these same different organisms are close relative. Suppose, for example, the first one is for human, suppose hemoglobin a segment. Second one is chimpanzee hemoglobin as first initial some few amino acids. Third one is your orangutan. Fourth one is your suppose gorilla. So they are all close relative. So if they are closely relative, if their protein is same hemoglobin, they must have the equal length, right? So if they are not having the equal length, then also their sequences may be more or less similar. So to analyze these four sequences, first criteria is to get their length equal. Okay, so length, making length equal, and by careful observation, by inserting gaps or substitution, we may get a better alignment like this. Just for example, okay. So if we align like this, so you can see that in second sequence, the second position, third sequence in the second positions, there is a gap, there is a deletion, we can say biologically. Deletion for what? This could be phenylalanine, right? F should be there, but F is not here in the second and your third sequence. So here your glutamic acid, E is for glutamic acid, right? So E is missing here in the second sequence. But still, by doing this alignment, we are getting the maximum number of conserved residue. This MMPP, HC are all conserved throughout, right? A simple naked eye will give you a lot of idea which cannot be detected. So sequences may be related by evolution, although we cannot detect it uh, in simple way unless and until you analyze the sequence in by applying some alignment procedure. Okay, comparison of the sequences. So why we should compare? So before comparing, we should ask ourselves itself that are these sequences are related, whether they are from evolution happens from the ancestor, common ancestor. Can we qualify their similarity? If they are similar sequences, so how much means what is the quality of the second? Suppose first is a sequence, second is another sequence, and we are saying that means I am saying that two sequences are similar but not identical. So can we qualify them? So this should be another question. And thirdly, we have to ask ourselves itself: do they have similar segment? Okay. So these are the basic questions should come before comparing the sequences. So some terminologies you need to have in your mind that when we say homology, the term common ancestry should come. That be careful that we are talking about sequences. Okay, we are not talking about organisms. So we are not talking about the two organisms are homologous or two organs are homologous. Not like that. We are talking about sequences. Two sequences are homologous. That means they are evolving from the common ancestor. Sequence or and usually structure conservation means their sequences are more or less most of the residues or most of the nucleotide are conserved throughout the alignment. And homology is not measurable quantity, so we cannot quantify the homology. We cannot say two sequences or three sequences are some percent of suppose 80% or 70% homologous. We cannot say that. That is a wrong statement. Okay, so homology can be inferred under suitable condition. So we can say only two or three sequence or set of sequences are homologous or not. They are homologous sequence or they are not homologous sequence. So that this term can be usable. We cannot quantify them. Next is identity. So we can say two sequences are identical or the three sequence or a set of sequences are having some identity. So what is the identity? So it is a objective. It is well defined and can be quantified by several methods like percent identity method, a number of identical matches means exact matches divided by length of the aligned region. Okay, so we can calculate the identity, we can give a score that four sequences are, we'll just find out the identity of these four sequences or two sequences. So you can identify, you can calculate the identity with a score. And similarity, see this terms looks very simple, but have very in-depth meaning. Okay, similarity. When we say similarity, most common method used. So these are most commonly used method, but not so well defined as identity, and depends on parameters used. So what is the parameter used? Uh, so you have heard the name leucine, right? Leucine is an amino acid, and isoleucine is another amino acid. So can you find the similarity between leucine and isoleucine? If you have chemistry background, then it's very easy for you to find out the similarity. What part is similar, what part is not similar between leucine and isoleucine. Okay, so that alanine and serine, to say alanine and serine are similar, we need to find their physicochemical properties or something like that. So similarity is somewhat a tricky point here. So it can be calculated by a kind of tables called scoring matrices. We will see after this slide, that is. So matching versus similarity, what is the simple understanding? So see, two sequences we have. So both sequences are same, right? So both sequences are not having any mismatches. They are 100% identical. So we can say that sequences are identical with 100% sequence identity, right? In this case, you see there are some mismatches. 1, 2, 3, 4, 5, 6, right? 6 mismatches are there. Alanine is substituted as serine, leucine as isoleucine again, then asparagine to your aspartic acid, then arginine to lysine, threonine to arginine, then arginine to lysine. So this, how will you calculate this similarity and identity? So these green colors are all matches. So total number of matches, right? So 10 matches, 16 the length. Total number of residue we have 16. So 10 are matched residue. Matching residues are number is 10. So you can easily find out the identity by percentage multiplied by 100. You will get 62.5% sequence identity, we can say. Now similarity, if we ask about the similarity, so we need to see some table. Okay, similarity usually calculated based on scoring matrices. So what is scoring matrices? Scoring matrices are kind of tables like this. Okay, the history of scoring matrices is very old, in 1960s. Initially, I think around 1962 or 1963, Day of Margaret and her co-workers developed this PEM, uh, initially 100, not 250. So PEM stands for point accepted mutation substitution matrix. So this is developed by a rigorous statistical analysis of alignment data. At the time of 1960s, protein sequence database SwissProt consists of only 1571 sequences of different sequences. So they have downloaded all the sequences and they have aligned these sequences manually to find out uh, what could be the score, means evolutionarily, statistically, what could be the score if we have to align, uh, by force tryptophan upon cysteine, suppose. So if in an alignment, say two sequence alignment, we have a sequence. The first sequence is one position tryptophan is there, and in the second sequence, at that position we have a cysteine. So if we have to align tryptophan with cysteine, what could be the score? It is -8. Okay. So if tryptophan is aligned with serine, it is -2. If it is tyrosine with cysteine, then it is -3. Okay. If tryptophan and tryptophan comes, if we have to align tryptophan with tryptophan in the second sequence, then the score should be your 17. See, the highest positive score. So this matrix will tell you a generalized understanding of molecular evolution. Okay, evolution means you can see those negative value. You can see, look into the negative values, and you just look into the positive values. Okay, where cysteine comes positive, when serine comes always positive, when theorin come it's still positive, proline comes then positive. So when there is no substitution, when two sequences are identical, we can give positive score. That means they are not mutated, right, throughout the evolution. They are not changing their amino acids, they are not mutating, they are not deleting anything. That means they are uh, conserved due to the evolution. So conservation is a favorable event in evolutionary process, right? So if something has to be substituted, two residues, tryptophan and cysteine comes, then it is C -8. That means you just look for some other values, 0, less than -8 or greater than -8, 0, right? So 0, C, arginine comes, proline. So no score, that is 0. So why it is -8? Suppose we are aligning two sequences and we are getting at a positions tryptophan is aligned with cysteine. That means that event, that substitution event, tryptophan is changing to cysteine, is a rarest event in evolutionary process. Okay, that is depicted by this kind of matrices. So nowadays many developed matrices are there in the software, uh, like block substitution matrices called Blossom. Okay, Gonet substitution matrices for nucleotide. So we'll be uh, coming across to those substitution matrices while using any tools or software. Okay, so BLAST is a tool by means of which we search uh, the similar sequence or homologous sequence or dissimilar sequence, either that is nucleotide or protein sequence. Here I have just shown one output of protein sequence BLAST. BLAST is okay. We'll be coming to that thing. BLAST is basic local alignment search tool. It is a kind of search engine for your sequences. Okay, search engine like Google, if you give this long sequence, suppose up to this 40 to 98, if you copy and paste over Google, Google will not be able to give you any relevant result. Okay, so you need to search sequence in BLAST of NCBI or any other BLAST maintained at any sources. Okay, so depending on the alignment types, depending on their uh, position, like where to align or how to align, there are two types of sequence alignment. One is called global alignment, one is called your local alignment. You see in the figure. So global alignment is an alignment where we compare two sequences from end to end, from the beginning to the end. We are aligning two sequences, concentrating on the matches or mismatches, whatever, from starting to the end. In local alignment, we will be concentrating on the local region matches. Local means, suppose for initial 4-5 characters, they are not showing any matches, that can be ignored. Okay, so if anything in between, they are very conserved, that can be identified by local alignment. So for example, genome alignment. Suppose human genome you want to align with chimpanzee's genome, preferably you have to go for a global alignment. If you want to align human genome with suppose some certain gene of human, it should be your local alignment. Again, protein sequence search against the database is local alignment. So These are the two basic categorization of alignments. So let us see again some text form. What is global alignment? So it compares sequences and gives the best overall alignment, global alignment. But it may fail to find the best local region of similarity, such as shared motif among distantly related sequences. So this second one is not the aim of global alignment, so it is failed there. Okay, so global alignment tends to align the sequences from the beginning to the end, not the local region. So local alignment will find regions of ungapped sequence with a high degree of similarity and better at finding motif. What is motif? We will I'll just elaborate it after a while. So especially for sequences that are different overall. So and it will only the best matching segment for a given pair of sequences. So it will return. So this is the best alignment when we perform searches like Google kind of search. If we want to perform for our sequence, that can be done with local alignment. Okay, so pairwise sequence alignment methods initially developed as dot matrix method. So in this method, uh, two sequences, pairwise sequence, two sequences are aligned. One is placed vertically, one is placed horizontally, and then the box against corresponding to them, you can put a star or dot wherever there is a match. Okay, you can see there is a match, means all I have placed both sequence same sequence. So I'm having one global alignment and two local alignment, right? These two are local alignment, but we cannot identify that global or local in dot plot method. Okay, that is the first and foremost disadvantage. And second disadvantage is it's very difficult to trace diagonal like this. Only and only diagonal will give you the aligned sequence. Aligned means matched characters. Okay, so if anything mismatch, or if we have to insert gap in some sequences, it will be problematic in case of dot plot method. So this programs comes to overcome those limitations over dot plot. So dynamic programming method is a computational method, computer aided method. So it is used in global alignment by using Needleman and Unz method, and Smith-Waterman we use for local alignment method. So this, if you need, we can explain in the upcoming classes, dynamic programming methods. So anything we use other than that, suppose online tools we are using. So online servers, online tools will use blast method or FASTA method. They are called word method or catapult method. Brief of your protein structures. In bioinformatics aspect, uh, we deal with protein structures in for prediction purpose as well as for visualization purpose. So let us see what we do in bioinformatics. So structure prediction motivation. Okay. So hundreds of thousands of gene sequences translated to proteins. Okay, they are already deposited in GenBank, SwissProt, or any primary databases. Then they are curated. They are placed in secondary sequence databases. But this is happening every time, every day. Thousands, hundreds of thousands of gene sequences are isolated, are submitted into the databases. If they are gene sequences, mm, the maximum chance that particular gene sequences may encode some protein sequences, right? So not all gene, but some gene are also for mRNA also. So, but maximum cases, gene, if you have that particular gene will encode a protein sequence. So that can be again translated by using computer. If you have isolated a gene sequence, you no need to isolate the protein encoded protein sequence again by wet lab techniques. Okay, you can do a conceptual translation of your gene sequence to get the protein sequence. Okay, so in this scenario, you are having day by day, you are having lot of data. It is increasing day by day, right? So for about it is I have taken this data 14th of February to 21, that protein data bank. Protein data bank consists of your protein structure data which are solved by experimental techniques, either X-ray crystallography or nuclear magnetic resonance spectroscopy or cryo-electron microscopy. By using these techniques, if structure is determined, that is called they are solved protein structures. So 14 February to 2021, I have recorded that the structural database is consisting of 1,74,507 structures. So do you think this much of structures are enough corresponding to the entire organisms in the earth? Sometimes I may ask you simple questions. You may respond so that I can get a feel that you are all listening. Are you all there?
हाँ सर,
Okay.
प्रेजेंट है।
Okay. So 1,74,507 solved protein structures means they are known structures in the protein data bank. So these are certainly not enough if we consider the entire organism's genomes on the earth, right? So some of the proteins are still structures are not determined. Experimental method are time consuming and not always possible. For example, most of the transmembrane protein, okay, you know transmembrane proteins, efflux pump, potassium pump, calcium pump, they are all proteins. Okay, ATP binding cassette superfamily GPCR, all are membrane proteins, membrane bound protein. So membrane bound proteins are very difficult to be crystallized and thereby very difficult to use X-ray crystallography for determination method. Even they are not soluble in solutions, so it is again not possible for NMR. So experimental methods are not always possible and they are certainly time consuming. So goal is, if we need to infer some function of protein and that protein structure is not determined, we can go for a prediction approach by using bioinformatics tools. Okay, so see scenario, one scenario, uh, I have shown an alignment, pairwise sequence alignment of two oxygen transporter. Okay, one is from human hemoglobin alpha chain, second one 2-HSDA, you can see, and one ECD, first one. You see two sequence alignment I have performed, and they are not sharing very good, conservations. Okay, alignment is poor. Match is we are expecting more matches because they are oxygen transport, and they are kind of hemoglobin. One is hemoglobin, and first one is erythrocurorin. Curorin, have you heard that? Erythrocurorin is a protein, another protein, kind of similar function as hemoglobin. Okay, heme may not be there in some cases, heme moiety. Okay, so these two sequences are dissimilar sequences, yet they share same structures. Okay, the your right hand side you can see that is hemoglobin, human hemoglobin, and your left hand side you can see it is a erythrocurorin. So erythrocurorin and your hemoglobin are similar structures, but their sequences are dissimilar. Okay. Another example, you see, I have taken some globin family of proteins alignment. They are from globin family, so we are expecting more matches in certain positions, but not even in certain positions. We are getting Very far locations we are getting one leucine, one proline, one phenylalanine and one histidine only conserved. Not at all other residues are conserved, not matched. They are all mismatches. Okay, they are from globin family. So they, even though they are from the same similar kind of no categories, their sequences may differ, but yet they can share the same kind of structures. So which makes the prediction approaches very difficult. Okay, from we now cannot make a rationale that okay, similar sequence may give similar structures. No, as we can see, even dissimilar sequences also share similar structures, erythrocytonin and your human hemoglobin. Then different structures may give different functions. Uh, okay, it may give, but different sequence may give different structures. That may not be true because globin family of proteins, the dissimilar sequences, but they are almost more or less they are alike. Okay, so we deal with uh, computers. Okay, there are prediction approaches, two stages initially. First, maybe primary sequence, two-dimensional sequence can be predicted to have secondary structures. Then secondary structures can be arranged into a tertiary form. Or more sophisticatedly, we nowadays we have more advanced tools or more advanced web pages, online tools. Only one stage, you just give the linear two-dimensional polypeptide sequence, you will get the tertiary structures. Okay, so according to most simplified model, in first step the secondary structure is predicted first of all based on the amino acid sequence. Then the secondary structure elements are arranged to produce the tertiary structure, that is the structure of the protein chain. Then for molecules which are composed of different subunits, the protein chains are arranged to form the quaternary structure. Okay, so let us see the protein tertiary structure prediction methods. What are the methods used for predicting uh, structures? So prediction methods are different from your determination method. Okay, protein structure determination, I am repeatedly telling that their determination method X-ray, NMR and cryo-electron microscopy. Determination method. Once you use this three method, you will definitely have some photographs or some files to be submitted into databases. But in prediction method, you will have again a similar kind of files like in determination method, but you cannot deposit into databases. So predicted structures of protein uh, nowadays cannot be accepted, means it's not been accepted by uh, your databases. Prediction is prediction may be correct or prediction may have some errors. So no databases are responsible to acquire the deposition data for their own guarantee. Okay, that is why prediction data are not to be deposited. Prediction data are to be for are for only you to furnish further analysis. Okay, so before predicting protein sequence data, you need to first identify which protein sequence you want to predict the structure. Okay, so obtain that sequence means just select that sequence or identify that sequence. Definitely when you target some project for protein structure prediction, definitely you have to do some prior literature review, literature search to find out on what area you are exploring. In that area, how many protein structures are determined. If something is not determined, or what? In another scenario, you may get okay, some pathway is happening that some ligand is binding to certain proteins and it is inhibiting that particular protein for curing that disease. All right, so then you are looking that particular protein into structure database, whether the structure is available or not. You got that structure is not available, so you can directly consider that particular protein sequence as your target for predicting your model. Okay, once your target is fixed, that okay, you have decided that this is the protein amino acid sequence you are going to predict the three-dimensional structure. Your first task is to assign the folds. Okay, there are different fold of proteins. For example, when a protein consists of only alpha helices, it is called alpha fold. When a protein consists of alpha helix and beta sheets distinguishably, then it is called alpha and beta fold. And if a protein consists of only beta sheets in a net-wise manner, it is called beta fold. If a protein is consist of beta sheets in such a way that it forms a barrel kind of structure, mostly in transmembrane proteins, is called team barrel. Okay, so I have listed some of the folds. There are many folds over the net. Okay, internet. Folds are recognized, folds are classified by specific databases by human curator. Okay, so we no need to worry about this is a procedure. You just have to have some idea how it is done. Software is there, software is enough for us to assign these kind of things, fold assignment and all. Just you need to know the steps. Okay, fold assignment will be done by software. Either you use offline or you use online. This can be done automatically, but you need to give the instruction to the software that okay, now you do fold assignment. Okay, so just you need to understand that there are different kinds of fold. You see, beta lactamase is a fold also. Beta lactamase is an enzyme, right? So you, all of you know beta lactamase. So beta lactamase has got beta lactam ring along with alpha helices and beta sheets. So this alpha helix beta sheet sandwiched by two alpha helices in beta lactamase. This kind of fold is common to most of the proteins, which is called beta lactamase fold. Same flavodoxin is a single protein. You can see the ID I have mentioning here there 4FXN. 4FXN is protein data bank ID. If you go to protein data bank and type search box 4FXN, you will get the flavodoxin entire protein. But this is what I am showing you is flavodoxin fold. Only the fold, because of which it is called flavodoxin. Okay. So fold assignment, once fold assignment is done, okay, you can look for you can look for some known structures related to your novel sequence. Novel sequence means your target sequence. All right. So if your target sequence, if you have done a database search like BLAST, basic local alignment search tool, to see whether any sequence present related to this sequence, your sequence, and having some structure available for that sequence. If structure is available and this sequence similarity of that structural sequence and your unknown structure sequence is suppose more than 60% or 70%, then we can go for searching a template. Template means a protein data bank structure, a structure already determined for that sequence, which is showing more than 60% of similarity to your sequence. Okay, if you find a structure like that, that structure is termed as template. So if you find a template, then you can align your sequence with the sequence of the template. All right. So once it is done, then you can build a model similar to the template, and after building you need to assess your model. Assessment is nothing but your uh, validation, like how much your predicted model is accurate in comparison to the already determined structures by determination method. Okay, so this kind of modeling, this kind of prediction is termed as your comparative modeling. Okay, why comparative modeling? Because you are comparing your sequence to find a known structure. Okay, already known structure in the database. So once you find it, then you can align your sequence with the known structure. So that is why it is called comparative modeling or homology modeling. You are modeling your sequence by comparing with a template, or you comparing with a homolog present in the structural database. Okay, so if this is a case of more than 50, 60 or 70% sequence similarity, if you get with the sequence of the template, so for suppose you don't get it, suppose you are not getting that much of sequence identity, then first criteria is to split your sequence into several small pieces and obtain a library of known folds. Library is not book library. Library of known folds means a collection of files containing the known protein folds. Obtain a known folds, then you can align your small piece to the all known folds, and you can finally trade them, trade the target on different folds to obtain a compatibility score. Okay, so once you traded it, no, you just stitched your all split sequence with the aligned small folds, then also you can build your model. And whenever you build your model, you finally have to validate your model uh, with the already known structures, how much accurate your build model with the structures available in the databases. So this kind of, again, modeling, this kind of prediction of protein structure is called your fold recognition. You are recognizing, you are finding the folds first, then you are stitching your sequence, right? Or it is called threading also. Fold recognition can be also sometimes called as threading. Okay, third approach is most complicated approach, in case of which you will not get a single similarity of small pieces also, and 50-60% you just forget it. You suppose you are not getting that at all, then assuming the criteria of the primitive assignment, like assume the native state of the protein is at global free energy minimum. Assuming this, with the properties of amino acids which are responsible for forming alpha helix or beta sheets, or which are the residues having the tendency of, excuse me, there is a network issue. Okay, okay, so oh, depending on their property to make their different secondary structural elements, uh, and assuming that structure of the protein is at global energy minimum, uh, we need to start with the basics. Okay, search for a tertiary structure that are low in free energy, and then finally build model and assess model, and this is called your de novo modeling or ab initio prediction. Okay, this part when comes, no, when you are getting your sequence, when you have target is fixed, when you have fixed your target, when you have assigned your folds into your target, and then you suppose uh, not found 50 for 60% similarity, if you not means if you are not finding any small fold similarity, then going to this method, performing this method is very error prone and sensitive. Okay, you need to be very careful for applying this method, ab initio prediction, and we mostly do not go for this if we uh, don't get significant amount of sequence similarity in these two cases. We don't go for structure prediction directly unless it is not very required. All right, so tertiary structure prediction tools we use mostly as online Swiss model for practical demonstration, and for research purpose there are many tools, mostly in Zhang lab from China. There is a tool I have not listed here called iTasser, and it needs academic email ID. .com will not help you. So .ac.in or .edu they require. Just only you need to submit your sequence, amino acid sequence. You need to submit into that server. So there would be a box where you can submit. Means you can copy and paste the amino acid sequence, and you can just click on submit button. So there are a lot of users, people are doing the same thing at the same time. So depending on their queue system, it will show you how much time will be required to deliver your output. So almost around 200 residue amino acid will take 24 hours time to be predict for structure prediction in case of those kind of ab initio modeling server. Swiss model is very user friendly, and it is based on your comparative modeling. You cannot go for uh, fold recognition or ab initio approach. Only homology modeling can be done by Swiss model. Okay, so predict protein server is another one. I think okay. Then modeler is an offline. Uh, it's a very interesting software, but for the layman like you may find something twisted or screwed up by using modeler, because you will not get any user interface. Graphical user interface is not available for modeler. But alternatively, there is something called easy modeler, which is having graphical user interface, and you need to install GUI and modeler both for easy modeler uses. Okay, so modular, otherwise, if you are very easy, you are very familiar with the command line, things in Windows or Unix operating system. So modular will be very helpful for predicting protein in any approaches. You can use threading, you can use ab initio modeling, you can use homology modeling. Okay. Apart from this, no modeling tools, you may require some validation tools also. Validation tools like how to produce result by means of which we can check whether our predicted model is close enough to the already determined structures or not. So they are either generating Ramachandran plot. One plot is called Ramachandran plot. We will see that. What is that? What is it? Uh, Project is used for generating Ramachandran plot. Then Verify 3D is one, another online tool where users need to upload your predicted structure. And what if is another validation tool where also you need to upload your structure, and it will give you some scores. So let us see some practical things, like experimentally determined protein structures can be retrieved from this protein data bank. Okay, you type by search box by name, or if you know the ID, you can type the ID. Okay, we will see how to get the IDs and how to search it, not today but upcoming classes. Okay, so molecular representation is another important aspect in structural bioinformatics. So molecular representation, this is one molecular representation of sperm whale myoglobin. Okay, you can see lines, some wire format. These are all wire format, right? These are all wire format, and heme is at the pocket of the myoglobin. This is one representation. Another representation could be your these thicker lines or ribbon format like this, or finally we may represent the entire things in molecular surface to see where is the pocket. So in molecular surface we can see the heme is quite compact in the pocket, right? In the pocket of the myoglobin. So that is the heme pocket. Okay, only molecular surface representation will tell you the where is the protein active site pockets, where is the surfaces, where could be some holes that can be viewed in protein structure, sorry, molecular surface representation. The application of bioinformatics is finally coming to the portions where we can apply bioinformatics. See, till now we have already discussed that we can perform sequence analysis and structure analysis. So in sequence analysis, what we can do, we can compare two sequences, pairwise sequence analysis. We can compare multiple sequences by multiple sequence analysis, right? We can generate phylogenetic tree from multiple sequence or sequence analysis. then we can have nowadays next generation sequence data and data, what we also can analyze by using bioinformatics tools, right? So in structures, let us see uh, structure prediction, right? We can just we have a look on this, how to predict the structures. Then we can predict protein-protein interactions. We can predict protein-ligand interactions. We can study uh, the dynamics, protein dynamics. Protein structure is determined, means we will have a picture, right? We photograph of a protein we will have. That photograph is not a dynamic photograph, that is a static photograph. Some state of the protein is crystallized and X-ray is passed, we got the X-ray crystallographic image, electron densities of the protein structures. So in actual scenario of the physiological condition, protein is not static. All of we know that protein is fourth dimensional in nature. Time dependent changes should happen in protein structures, right? So those time dependent changes can be studied in computer, and they are called protein dynamic studies. Okay. Similarly, in protein-protein interactions, in wet lab techniques, what you are familiar with, protein ligand interactions, protein-protein interactions, you are familiar with immunoassays, right? Precipitations, yeast to hybrid method. These are wet lab techniques. So two proteins are interacting, precipitations you are getting, that means okay, they are interacting. So these kind of things we won't do. We will not do in computer. We will directly take two protein structures and we will calculate their binding energies. We will take either protein or ligand, we will calculate their binding energies at the binding pocket. With those laws, with those thermodynamic laws, with those Newtonian laws, which are there, which we have already have gone through in 10+2 books. The same laws will be applied in an integral way in all atoms upon time. All right. So protein ligand interactions and protein dynamic studies are rigorously used, rigorously used in a separate field called computer aided drug design. So you don't think that computer aided drug design will give you a drug or molecule or a capsule coming out from your computer screen. Okay. So computer aided drug design is a aided tool, is a helping hand for the actual drug discovery pipeline. Okay. So molecular dynamic simulations are used for protein dynamic studies. You can start with an initial structure from protein data bank. You can identify their potential energy at the very beginning, and then you can apply some force, minimal forces depends on means your study. If you are studying the protein in a test tube, for suppose in a test tube at some certain degree Celsius with some solutions, and if you are hitting for denaturing the protein, suppose, so that can be defined as a force. Okay, we need to apply those heating laws, law of heat, no, specific heat. So ultimately numerical integrations can be done to get a trajectory. Trajectory, what is it? Is a time no time dependent uh, movement of the protein. Okay, is called your trajectory here. All right. So equations flow of the equations will be looking like this. So no need to afraid of. We are not going to deal with the mathematical portion of any of this softwares. Softwares are already built. We need to know the uses, their uses. That's all. Only to make you out of these subjects. I just have a just slide for representation of molecular dynamics simulations. So simulations are useful for, suppose this is the whole structure of our recent pandemic coronavirus. Okay, so these structures can be visualized like this with a movement by using molecular dynamic simulations. So water molecules can be moved like this. So these are water molecules. You can see red colors are oxygen and white color are hydrogen atoms. So we can define a box like this, and box should be big enough to accommodate the protein or a virus, whole virus, viral capsid. And then suppose for again for transmembrane protein, membrane as well as the protein within this water molecular box. Then finally we can have a movement up to certain time scale. The time scale should not be hours or days. Okay, these are microscopic molecule, even ultramicroscopic molecule. We cannot see these molecules under microscope, right? So these are ultramicroscopic or supra microscopic molecules, and time dependent changes, time we need to consider for these molecules should be around less than seconds. Okay, after suppose uh, a few milliseconds, what would be the positions of certain water molecules, or a kind of, suppose this is a transmembrane protein simulations with membrane, we have hidden the water molecules, so drug is bound at the middle, red color in the protein molecules. So what would be the fate of the drug after some time? We can calculate by molecular dynamics simulations. Okay. Next, we can use protein-protein docking, protein ligand docking for finding the affinity of the small molecule or a drug at the binding pocket of a protein. For example, there are many HIV protease inhibitors, right? And many some protein inhibitors are there. So that how they are inhibiting that protein in wet lab experiment, you need to find out the either cytotoxicity against some cell line, or maybe some what to say, some wet lab experiment only. You need to find out IC50 values or something like that to find out the toxicity, or whether that particular compound is or molecule is inhibiting the protein or not, inhibiting the entire cell line or not. But in docking, in protein ligand docking in computer, we will find out the best binding position configuration of the ligand at the binding pocket on the basis of their binding energies. Okay, so extremely uh, mathematical task is going on uh, at the back end of the softwares. Okay, so dynamics of the molecule is making everything difficult for us. Us means computational biologist. Okay, this is a model called your molecular mechanical model. What you are observing now, uh, three bonds. No, one bond is stretching here and there, one is rotating, and another is your flipping, right? So this kind of model representation for dynamic behavior is called your classical mechanical model or molecular mechanical model based on Newtonian laws. Okay, based on Newtonian law, we cannot calculate the new bond formation or existing bond deformation or destruction. We cannot define, we cannot observe a new bond formation or existing bond destruction. We cannot see in molecular mechanical model. Okay, even then also this is very again expensive, computationally expensive. You cannot perform this kind of, this is a GIF image, okay, GIF image, what you are observing. Suppose if I uh, uh, assign the attribute that what you are seeing, no, at the terminal positions they are hydrogens, and then red color are oxygens, blue color are nitrogens, then what I have to assign their atomic numbers also, right? So if I assign their atomic numbers, for this entire molecule, I will be having around 15 line or 20 line of data. So all these 20 row of data I have to simulate. It's not a big task in molecular mechanical model, because we are having some good laws, physical laws for those frequencies, laws and all, stretching, omega things. So it's very easy. But in order to observe a bond formation or bond deformation, we need to see in this figure. You see one molecule, one atom, certain atom of a certain atom will be detached from here. This atom will come and join somewhere to form another bond. See, it is joining to this one and forming another bond there, right? So this kind of molecule atom dissociation and forming bond will not be calculated by classical mechanical classical mechanical model or Newtonian mechanical model. We need to deploy another thing is called quantum mechanical models. So you see, that's a molecular orbital theory will come when you deal with your quantum mechanical model. So we cannot consider just by bonds, simple bonds. We have to apply all those forgotten laws, Pauli's exclusion principles, then Heisenberg's uncertainty principles for quantum mechanical models, because here bonds concept of bond is different than your classical mechanical model. Okay, so here methane or ethane is looking like this. In molecular mechanical model, we can assume just ball and stick model. You just assume double bond is here, is a spring like properties, is a molecular mechanical model. But in orbital theory, you have to consider like this, electron density kind of things will come, and it is more and more complex phenomena. Okay, in actual drug discovery pipeline, where bioinformatics can be useful. Okay, let us have a simple gist about that. From identification of a disease to the FDA approval of a drug. Okay, let us see, in traditional way, if we do not use any computational tools or any bioinformatics softwares, any bioinformatics method, then let us see how much time will take from identification of a disease to get a new drug approval. Okay, so identifying a disease, what may be causing the disease? It may be a causative thing is maybe definitely a protein. Either protein is malfunctioning, either protein is not expressing properly, or protein is suppressing, or that particular protein is not at all producing in our body. So that may be the cause of your, suppose, the protein. So for isolating, to get the knowledge that okay, this is the protein which is causing that particular disease, it will take around maximum of 5 years, right? 2 to 5 years. So you just consider the upper limit. Just consider 5 years will take for the finding of the causative protein. Then find a drug effective against the disease protein. Against this particular protein, which drug can be effective? That finding will take another 5 years. So 5 plus 5, 10 years, right? So after getting this one, no, after this is okay, this is the drug effective, this is the effective drug, then you cannot directly apply on human, no, you have to uh, apply in animal models. Preclinical testing will take another maximum of 3 years. Upon successful completion of preclinical trial, you have to apply for production to a extent, a large scale production you need for this molecule. So you need to file and scale up and formulation, you need to prepare for that. And once you scale up and formulate it, you have to file an application to FDA, that file an IND. IND means investigational drug administration. Okay, you are investigating a drug administration, means you are trying to administer a drug on human, but before that you are doing some preclinical trial, and because of which you need formulation as well as scaling up. That is a huge number of production you want. For that you need to take a permission from FDA, that is called investigational drug administration filing. So once you are approved for that, you can go for, means if you are successful in this, no, if you are successful in preclinical testing, then you can go for human clinical trial, which is most time consuming part. You see, 5 plus 5 plus 10 plus 3, 13, and upper is your 10. Okay, it is unfair if we take 10. Okay, let us take in between, suppose I get another 5. Okay, 5. So 5 plus 5 plus 15 plus 3, 18 years. Then FDA will not approve, no? If you are successful in human clinical trial, your drug is successful, then you have to file, you have to apply for this drug to approve in the market. Then FDA will take another 2 to 3 years, which is totaling all of about 20 to 22 years. Okay, so a new drug to be launched in the market will take around 20 to 22 years if you do not use any bioinformatics approach. Okay, if you use bioinformatics approach, where to use? We have to use in this. Okay, so finishing. Okay, this is the last slide. So we can use bioinformatics here itself, that identification of the disease, isolating a protein, how to isolate a protein. We can outsource it. Genomics, proteomics, and biopharmaceutical companies potentially producing many more targets and personalized targets. Okay, for a disease, you just state them. Okay, for this disease, you find some targets protein which are not already established. So they will do it for you, or you can invest some time faster than ever. But just if you can, just give some time and just find in computer search. Okay, genomic proteomics biopharmaceutical companies, we are usually doing it. Okay, then high throughput screening can be done by computer, screening up to 1 lakh compounds a day for activity against a target protein. Okay, first well screening by computer we can perform using a computer to predict the activity of the 1 lakh compound. Okay, then combinatorial chemistry is a wet lab technique we can use. If we are filtering, suppose from 1 lakh compounds, we are filtering by virtual screening, we are getting 5,000 compounds, or we are getting 500 compounds. Those 500 compounds uh, we can modify to get more, same active compounds by using combinatorial chemistry approach by modifying its side chains or functional groups. Okay, so once we can get more molecules, more compounds, then we can use again computer graphics and models to help to improve the activity of the molecule. Okay, then before performing in vitro or this animal model testing, we can we can again filter the compounds based on their ADME properties. ADME properties are absorption, distribution, metabolism, excretion, and toxicity. It is also to be called as ADMET properties of the molecules. Means a molecule to be said as drug if it is going to be properly absorbed by our body, distributed into our bloodstream, metabolized throughout nicely from our body, and excreted throughout the body, and it will exert no toxicity to our body. Okay, so this ADMET screening can be again done by computer. Okay, if you are filtered out again from 1 lakh to 500, 500 to 100 by combinatorial chemistry, you are again increasing 100 to 200, then you can screen 200 compounds by ADMET properties. Okay, from 200 compounds, suppose 150 compounds are showing very toxic, and they are lessly absorbed by our bodies, and they are not properly distributed into our bloodstream and not even properly metabolized. So we can ignore 150 compounds. 50 compounds are passed in ADMET trial, and this ADMET compounds, 50 compounds, you can suggest for preclinical testing. You can inject these compounds into the mouse or animals for preclinical testing. So here we can use the bioinformatics approach, and this concludes the today's introductory note on briefings of bioinformatics. So from tomorrow onwards, you will be getting one by one, you will be getting the introduction to the application of the bioinformatics. So any doubts you can clarify? So if you have doubt, you can ask. Okay, no issues, no problem. So how about you people? You are all engineers, right?
No, sir. I am from biology department.
Good. Okay, so all are from some diverse discipline. So
Yes.
just give a feedback, or if you have some doubt, you can clarify, or if or it's you can leave, no problem.
Okay, sir.
Sir, are you there, sir? Saurabh, sir?
So where we will get this PPT?
Yeah, you will be getting this all. So let us design one, classroom, Google Classroom. So we'll be, uploading all the PPTs there. So you'll be all getting this everything from the classroom only. Okay.
Okay, sir.
This program will be continuing to another 9 days, right? This is the first day.
Yes, sir.
So, okay, so you'll be getting lot of notes, lot of PPTs, lot of materials. So let us get one classroom by today. So you'll be joining tomorrow within the classroom only with the same link. So no worries. Everything, every materials will be uploaded there. So you'll be all getting that.
And practical session will start from tomorrow.
Uh, let us see when we can finish the theories. So I think it will be divided as half and half. I think five will be theories or five will be practical. So I think that Saurabh Mohanto sir will tell you. Sir, are you there, sir? Saurabh sir, let me contact him. So he will be able to clear your query. ছাৰ এওঁলোকে ইয়াতে আছোঁ ছাৰ। ছাৰ কিবা সিহঁতৰ ডাউট মানে হেৰি আছে, কেতিয়া প্ৰেক্টিকেল হ'ব কি হ'ব সুধিছে। ৰিপ্লাই
Hello, hello. আছে নাই কথা আছে?
Yes, sir.
কোৱা? Yes, please speak.
when will be the practical now?
So practicals will be conducted in between the session itself, and we will give some assignment through the Google Classroom. Okay.
Okay, sir.
Okay. Thank you then. Thank you.
Thank you, sir.